Good day, everyone. It's me, Dryle. Today we're diving into an exciting topic: gene therapy. What exactly is gene therapy, and how does it work? We'll explore both its potential benefits and some of the risks involved. So let's get started and discover how this groundbreaking science is reshaping the future of medicine. Imagine a world where we could treat diseases at their very source, within our DNA. This is the promise of gene therapy. By altering the genetic material inside our cells, gene therapy aims to correct the underlying cause of genetic diseases, transforming how we approach medical treatments. But gene therapy is much more than just a futuristic idea. It is a field of medicine grounded in decades of research. And today, it offers hope to millions of people suffering from conditions that were once considered untreatable. To understand how gene therapy works, we must first understand DNA. Our bodies are made up of trillions of cells, and inside each of these cells lies a complete set of instructions that governs how the body grows, functions, and repairs itself. These instructions are written in the form of DNA, a long molecule composed of units called genes. Genes carry the information necessary to make proteins, the building blocks of life. But what happens when these instructions are faulty? A mutation in a single gene can cause a protein to malfunction or be missing entirely, leading to a wide variety of diseases, from inherited disorders like cystic fibrosis to cancers. Gene therapy seeks to correct these mutations by introducing new, functional copies of genes into the body's cells. It's like correcting a misprint in a recipe. Once the instructions are corrected, the body can once again function as it should. The origins of gene therapy trace back to the 1970s, when the field of molecular biology began to take shape. Scientists discovered a revolutionary technique known as recombinant DNA technology. This breakthrough allowed them to manipulate and combine genetic material in ways that were previously unimaginable. Recombinant DNA was the foundation for genetic engineering, where scientists could take DNA from one organism and insert it into another. With this newfound ability, researchers began to wonder, could we one day repair faulty genes inside human cells? It took nearly 2 decades for gene therapy to move from a theoretical concept to a clinical reality. In 1990, history was made when a 4-year-old girl named Ashanti De Silva became the first patient to receive gene therapy. She suffered from severe combined immunodeficiency, or CID, a genetic condition that left her defenseless against infections. The idea behind Ashanti's treatment was simple, yet revolutionary. Scientists would deliver a healthy version of the gene she was missing into her immune cells. In essence, they would reprogram her immune system to function properly. Ashanti's successful treatment marked a major milestone. It demonstrated that gene therapy could be safe and effective, even in cases where conventional treatments had failed. For Ashanti and her family, it was nothing short of a medical miracle. The process of gene therapy is based on a relatively simple principle. If a gene is malfunctioning, you can introduce a working copy to replace or repair the defective one. But how do you get the new gene into the body's cells? This is where the concept of vectors comes in. In gene therapy, vectors are the carriers that deliver the genetic material into the patient's cells. One of the most effective types of vectors is a virus. Viruses are naturally designed to enter cells and deliver their genetic payload, typically to cause infection. But in gene therapy, we modify these viruses so they can no longer cause disease. Instead, they are programmed to carry therapeutic genes. Once the virus enters a cell, it releases the new genetic material, which integrates into the cell's own DNA. From there, the cell begins using the corrected gene to make functional proteins. There are 2 major forms of gene therapy: somatic and germline. Somatic gene therapy involves altering the DNA of non-reproductive cells. This means that any changes made are limited to the patient and cannot be passed down to their offspring. Somatic gene therapy is already being tested and used for a wide range of conditions including blood disorders, cancers, and neurological diseases. On the other hand, germline gene therapy targets reproductive cells, the eggs or sperm, meaning that any changes made could be inherited by future generations. This type of therapy could potentially eliminate genetic disorders from entire family lines, but it also raises profound ethical questions. The idea of altering the genetic makeup of future generations is both exciting and controversial. On one hand, it offers the potential to eliminate hereditary diseases forever. On the other, it opens the door to more troubling possibilities. Could gene editing be used to select physical traits or intelligence, leading to the creation of designer babies? For now, most countries have placed a moratorium on germline gene therapy due to these concerns. But the debate is ongoing, and as technology advances, so will the discussions surrounding its ethical use. Despite the ethical debates, gene therapy is already transforming lives. Take, for example, patients with inherited retinal diseases, which cause blindness. In recent clinical trials, gene therapy has enabled these patients to regain their vision, something that was once thought impossible. By delivering a healthy version of the gene responsible for producing the proteins necessary for sight, doctors have been able to restore vision in patients who were once completely blind. These early successes are just the beginning, as researchers continue to explore the potential of gene therapy for a wide range of conditions. One of the most powerful tools in the gene therapy toolbox is CRISPR-Cas9. This revolutionary technology allows scientists to precisely edit specific parts of the genome. Imagine being able to cut out a faulty gene, like a pair of molecular scissors, and replace it with a healthy version. CRISPR is incredibly versatile. It can be used to treat genetic diseases, modify immune cells to fight cancer, or even prevent viral infections. The precision and efficiency of CRISPR have made it one of the most promising advancements in gene therapy, and it has already shown success in treating diseases like sickle cell anemia and certain forms of cancer. But gene therapy is still in its early days. Many challenges remain. Scientists are working to improve the safety of viral vectors, to ensure that the therapy is delivered precisely where it's needed without causing unintended side effects. Additionally, there is the challenge of making gene therapy affordable and accessible. For now, these treatments are expensive and complex, but as the technology matures, the hope is that gene therapy will become a standard part of medical care, available to everyone who needs it. As gene therapy continues to evolve, its potential applications stretch far beyond rare genetic diseases. Researchers are exploring its use in treating more common conditions like heart disease, diabetes, and neurodegenerative disorders such as Alzheimer's. The power to edit and repair the human genome opens up a world of possibilities. Possibilities that may one day change the face of medicine entirely. Gene therapy represents a new era in medicine. As we unlock the secrets of the human genome, we move closer to answers once thought unreachable. Treatments for the most complex and debilitating diseases that humanity has faced. That wraps up our introduction to gene therapy. I hope you found this information helpful and learned something new today. If you did, make sure to give this video a thumbs up, leave a comment with your thoughts or questions, and don't forget to subscribe for more videos on exciting topics like this. Thanks for watching, and see you in the next one.